Hello students, this is Dr. Ranjini, Assistant Professor in Biotechnology. Today in this session, we learn about amino acids. In nature, we have the polymeric molecules like carbohydrates, proteins, lipids and nucleic acids. The proteins are made up of monomeric units. They are called as amino acids. Amino acids are monomeric units. They join together by the bond, specialized amide bond called peptide bond, and they form the proteins or they form the peptides. So these proteins are having many functions inside the cell. On hydrolysis releases amino acids. The amino acid is bifunctional molecule having one positively charged functional group, that is amino group. The another functional group is negatively charged carboxyl group. It's a bifunctional molecule having the two functional group. One is positively charged and another one is negatively charged. It has the positively charged amino group and negatively charged carboxylic group. That is, both the functional groups are attached to the alpha carbon atom. They are attached to the alpha carbon atom. And this carbon is attached to another side chain referred as R group. This is the general structure of amino acid. Amino acids, they have alpha carbon atom, to which on one side we have the COOH group which is negatively charged. On the another side we have the NH3 group or NH2 group or the amino group, and you have the side chain that is designated as the R group, and the other end it will be attached to the hydrogen. This is the general structure of the amino acids. Amino acids are bifunctional having the acidic and the basic functional group, with one end hydrogen, on the another end it has the side chain or the R group. So this amino acid, under physiological condition, the amino acid will be showing both the positive and the negative charge. Otherwise, it will be having R, CH, COOH and NH2. So, this is the general structure of amino acid, where COOH is the carboxylic group, NH2 is the amino group, R is the side chain, H is the hydrogen atom. That is the general structure of amino acid. But under physiological condition, it will be having the NH3 plus, CH, R, COO minus. It has dissociable functional groups. One is carboxylic and another one is amino group. So this structure is called as Zwitterionic structure. This structure is called as Zwitterionic structure. What is Zwitterionic structure? It is the dipolar ionic structure of the amino acid. On one end it has the positive group, on the other end it has the negative group, and both the functional groups are attached to the alpha carbon atom. On the other hand, it is attached to the side chain, that is R group. They will ask you in the examination, write the general formula of amino acid, explain the terms. The general formula of amino acid is this. So, you should write NH2CHR COOH. That is Zwitterionic structure. NH2 is amino group, COOH is the carboxylic group, H is the hydrogen atom, C is the alpha carbon atom, R is side chain. And you will also have the question like explain Zwitterionic structure of amino acid. Then you have to show NH3 plus, CHR, COO minus. This is a dipolar ion of the amino acid, and this Zwitterionic structure will be observed in the physiological pH. So most of the in most of the condition we find the amino acid in the Zwitterionic structure. Isoelectric point of amino acid. Isoelectric point of amino acid, it is represented as Pi, is defined as the pH at which amino acids exists as the Zwitterion. What is pi? pi is the pH at which amino acid exists as Zwitterion or the dipolar ion. Its net charge on the molecular will be the neutral. So it will be having one positive charge on the NH3 plus, one negative charge on the COO minus. Both get cancelled and you will not have the any net charge on the amino acid at particular pH. That pH where it shows both the positive and the negative charge but no net charge on its surface, it is called as the isoelectric point. It is represented as pi. Because of the presence of the two dissociable group, we will be having the two dissociation constant. One is specific to the COOH group, the another is specific to the NH2 group. So you have the one dissociation constant specific to acidic group, the another one will be specific to the basic group. The pi usually will be the dissociation constant of acidic group plus dissociation constant of the basic group divided by 2. So one is acidic dissociation constant, the another one is the basic group, that is the amino group dissociation constant divided by 2. Sometimes if we have the side chain which is having the dissociation group, even that will be counted. So that is the another dissociation constant specific to the side chain or the R group. So this is regarding the dissociation of the ionizable group. One is specific to the COOH, the another one is specific to the NH2 group. So what is pi? Pi is called as isoelectric point. It is defined as pH at which amino acids exist as Zwitterion or the neutral molecule will not move towards the positive electrode or towards the negative electrode when you apply the electric current, because both the charge gets cancelled and it will be neutral. pi depends on the structure of the side chain of the amino acid also. So if the dissociable group is there in the R group, then also the pi depends on the dissociable group present on the side chain. The acidic amino acids, they have the isoelectric pH approximately 3. And basic amino acids, they have the isoelectric pH approximately 9. And neutral amino acid, they have the pi of slightly acidic, that is 5 to 6. So this is very important as far as your short notes questions are concerned. So this pi will be asked in the examination, maybe for 2 marks or for 5 marks. What is pi? pi is the pH at which the amino acid exists as the dipolar ion or the neutral molecule having no net charge on its surface. And this depends on the dissociation of the acidic group as well as the basic group. In addition, if the amino acid has the dissociable group on the R or the side chain, even that will be considered. And most of the amino acids which are acidic, they have the pi of the 3, and basic amino acid, they have pi of around 9, and neutral amino acid will be having the pi of around 5 to 6. And there are 20 amino acids. These 20 amino acids, they are present in different numbers and they constitute the protein. And these 20 amino acids are the constituents of most of the proteins that exist in the living organism or in any proteinaceous substance. And these 20 amino acids are called as magic 20 amino acid. 20 amino acids are called as magic 20 amino acid. Based on the requirement by the cell or the organism, these amino acids are classified into essential amino acids and non-essential amino acid. So amino acids based on their requirement will be classified into essential amino acids and the non-essential amino acid. Essential amino acids are amino acid. They are required by the cell for its function. These are required by the cell for its function. The cell cannot synthesize these amino acids. The cells cannot synthesize these amino acid that include arginine, isoleucine, histidine, leucine, methionine, lysine, phenylalanine, tryptophan, threonine and valine. So these amino acids are essential amino acid. These are not synthesized by the cell. They have to be supplemented through the food they take in. So these essential amino acids are very important for the cell functioning as they are not synthesized by the cell. They must be supplemented in the diet in order to obtain the normal growth or for cell to perform its normal function. The other amino acids, the non-essential amino acids which includes alanine, arginine, aspargine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, tyrosine. About 9 to 12 can be manufactured by the body itself, by the cell itself. Such amino acids are called as non-essential amino acid, will be synthesized in the cell. You need not to supplement through the diet. They are called as non-essential amino acid. About 9 to 12 amino acids are non-essential, will be synthesized by the cell. And these non-essential amino acid need not to be supplemented through the diet. But remaining around 8 to 10 amino acid has to be supplemented through the diet. They are called as essential amino acid. So we have 20 amino acid. So these 20 amino acids are classified into essential and non-essential amino acid. So essential amino acids has to be supplemented through the diet, whereas non-essential amino acid need not to be supplemented through the diet because non-essential amino acids are synthesized by the cell for its cell functioning. Next is pKa value of amino acid. pKa value of the amino acid is the negative base to 10, negative base 10 logarithm of acid dissociation constant of the solution. Acid dissociation constant of the solution. It is the negative log to base 10 of the acid dissociation constant of the particular amino acid. If the pKa value is smaller, amino acid is stronger. If it is more, then it is weak amino acid. So this definition will be used even for categorizing the strong acid and weak acid. Here also pKa is the negative log to base 10 of the acid dissociation constant of the amino acid. So pKa value given for the amino group on any amino acid specifically refers to the equilibrium between protonated positive nitrogen and deprotonated neutral nitrogen. It is the equilibrium between protonated positive nitrogen, NH2 gets converted into NH3 plus, NH2 will be converted into NH3 plus, whereas, so it is protonated positive nitrogen and deprotonated neutral nitrogen. So this defines the acid dissociation constant or the pKa value of the amino acid. Less of the pKa value, smaller the value of pKa, stronger is the acidic nature. The higher the value of pKa, the lesser is the acidic nature. Next is all amino acids in proteins, they exhibit the steric configuration of L-glyceraldehyde. That means amino acids we find in the nature all are L amino acid. Amino acids are L amino acid which are abundant in the protein. So these L amino acid, they have L amino acids, they have NH2 or the amino group towards the left side, and the D amino acids, they have the NH2 towards the right side. Amino group is always left side of the alpha carbon atom. Then such amino acids are called as L amino acids. They are present in proteins. D amino acids never found in proteins. You don't find these D amino acids in protein, functional protein, although they exist in nature. Although we have the presence of D amino acids in nature, they are not present in the proteins. These D amino acids are often found in polypeptide antibiotics. They are present in antibiotics, but they are absent in proteins. They are absent in proteins. So what is the difference between L amino acid and D amino acid? L amino acid, they have the amino group towards the left side, and D amino acids, they have amino group towards right side. And L amino acids are predominant in all the proteins, and the D amino acids are absent in the proteins. They are present in the antibiotics. So in this session, we learned the definition of amino acid, general structure of amino acid, Zwitterionic structure of amino acid, PA value of amino acid, then pKa value of amino acid, and few basic classification of amino acid, like based on the nutrition, the amino acids are classified into essential and non-essential amino acid, and based on the configuration, amino acids are classified into L and D. I hope you understood this session. Thank you.